Hi, everybody. Welcome to a reboot and the August installment of the Southern California Stem Cell Consortium. As you can imagine, we've been on a bit of hiatus, as has most of the world, particularly with conferences. But thanks to a lot of creativity from the organizers, and particularly Jake and Marcy, the idea of rebooting the Southern California Stem Cell Consortium virtually has now become a reality, and we thank a lot the hard work of UCTV as well, UCSD TV, in helping us do that. And actually, we've also been joined in this effort by the UCSD Alzheimer's Disease Research Center Neurodegeneration Seminar Series, and also help from Sanford Burnham Prebys and from UCSD. So one of the ideas that we had, and I give a lot of credit to Jake and Marcy for this, is the notion that this is a great opportunity to highlight hot papers in the stem cell field, and we define that exceedingly broadly. As a hot paper comes out, we can actually go to the investigators to talk to our audience about what the work was, what the thinking was, and where they're going from there. And the first participant in this is going to be Xiang Dong Fu from UCSD, who had an exceptionally impactful paper that just came out recently entitled Reversing a Model of Parkinson's Disease with In Situ Converted Nigral Neurons. Clearly going to be perhaps a transformative paper, and given that Dr. Fu is right here on campus, it was a no-brainer to have him participate and kick off this, this new version of SoCal Stem Cell Consortium. I don't want to take too much time away from Dr. Fu, just to say that he really is a homegrown product of which we're exceptionally proud. He did his PhD at Case Western Reserve, then did a postdoc with Tom Maniatis at Harvard, and really has been one of the thought leaders in RNA biology, particularly non-coding RNAs and RNA binding proteins. And as you'll see, has been taking advantage of his insight into that field to understand development, cell fate switch, and various aspects of disease progression. In his training years, he was a Searle Scholar. He's a fellow of AAAS, and worked his way all the way through the ranks at UCSD, starting as an assistant professor and now as a full professor, and was co-director of the Genes and Genome Program at the Morse Cancer Center. So this is it's a real pleasure to introduce both a colleague and a friend and a coworker working in a very important field, talking about a very important recent paper. So, Xiang Dong.
Thank you very much, Evan. It's indeed a great pleasure to participate in this new series of seminars, and of course we have to overcome the current problems, but I think none of us are stopping our academic activities. So today I would like to share with you a journey that we have been taken for more than a decade, almost 15 years from now, from basic science without any clue how those discoveries or scientific research might be translated someday into a real disease fighting process, to a discovery that may open a new door for us to understand and for the treatment of neurological disorders. All right, so this is my title, which is also the title of the paper, Reversing a Model of Parkinson's Disease with In Situ Converted Nigra Neurons. I'm from Cellular Molecular Medicine, UCSD School of Medicine. begin with the big picture stories. This is the quote from an article 2 years ago, 2018, by Zevan, oh my goodness, how to pronounce the last name, who is the former director in the Office of Alzheimer Research at the NIH, responsible for coordinating all AD-related activities. In this article, he used the title 40 Years of Alzheimer's Research Failure, Now What? A decade-long odyssey with little to show. What he highlighted in this article, aimed for general public, is that 40 years after intensive research and development effort has most of the time failed to yield any effective intervention for neurological disease, AD included. This failure rate is almost 99.6%. Probably he means one of them has some partial efficacy, which compares with about 20% successful rate for cancer drugs. As we know, 40 years ago, by the way, Richard Nixon declared war against cancer, but 40 years later, now we have a major breakthrough in cancer immunotherapy and many other advances that many cancers become manageable. But now we are facing a bigger challenge in understanding the aging as well as age-related disease. The problem is because of the failure, pharmaceutical company, many of them have stopped their effort in R&D and on AD. Many may follow. Thus, the question is that we really need new thinking or breakthrough in these disease areas. So specific for Parkinson's, there are many clinical features, but mainly for most people who are familiar with the Parkinson's, it's difficult to make a move. This is controlled movement, as well as the loose control for unwanted movement, frequently referred to as a tremor phenotype. So this is a typical Parkinson disease patient. And we know Parkinson is one of those diseases with relatively clear cause, which are related largely to so-called the impaired nigra striatum pathway, which consists of a dopaminergic neuron originated in the midbrain on top of the brainstem, and then targeted the axon to a region called striatum, where the neuron released dopamine to trigger the next neuronal activities, eventually control the motor neuron in thalamus. As you can see at the bottom on the left, the normal brain, you can imagine this L-DOPA biogenesis and actions, but in the Parkinson's disease patient, these dopaminergic neurons are dispersed and died and diminished. There are many ways to treat neurodegenerative disease. The first layer being symptom management. But that one, this type of approach has been extensively applied to Parkinson's disease patients, such as the oral take of L-DOPA, deep brain stimulation, so on and so forth. One of the major ideas is to condition the neuron with neurotrophic factors, or express or reinforce or boost the production of critical enzyme in those neurons. A third one would be the block anti-disease genes. Many of them has been identified as pioneered by also experts in this campus down Cleveland. But however, the problem is that many diseases do not, genetic components for many of those neurodegenerative disease can only explain about 5%. A large of them are sporadic, so-called. Okay, and most of those diseases are associated with formation of toxic aggregates, so effort has been made to resolve or prevent the formation of such an aggregate. Unfortunately, the approach developed so far showed little clinical benefit, likely because it was already too late, probably. by the time you diagnose it, and then all those symptoms developed, may be too late to intervene with that strategy. And of course, prevent neuronal death, which is also ideal. But the problem is that in those neurodegenerative disease, neurons become dysfunctional before death, long before become dysfunctional long before death. So the final approach, which is the new hope, which is to replenish lost neurons to just repair the damaged brain. And then this approach is only a decade long, which started with actually the new discovery of iPSCs, in the other word, generate stem cells, and use the stem cell to differentiate into different type of neurons, which can be implemented or grafted into the human patient. So this is why we have this stem cell program, which many active research being pursued in these areas. But related to this stem cell idea is called transdifferentiation of long neuronal cell to neurons. This is a little bit even younger, only a few years in research, has not been extended to a large animal model yet. But the advantage of this transdifferentiation strategy has many as we will go along. So with this general introduction to the disease mechanisms, disease interventions, I'd like to start to share with you how we stumble into this this course of research and discovery. This is because many years ago, we're talking about dated to 2003. So that's why we're talking about more than 15 years later. We were trying to optimize a technology in the lab called CLIP, cross-linking immunoprecipitation. As Evan introduced, I've been working on RNA biology through most of my career. And then this is one of the key techniques that I have highly useful, developed by Bob Darnell from Rockefeller University. The procedure is diagrammed here in this slides, that you take your cell or tissues, disperse the cells from the tissues, treat that with UV to induce cross-linking between protein and RNA, and then you can use your antibody against your favorite RNA binding protein to IP or immunoprecipitate your RNA binding protein and trim the associated RNA down, permit for linker ligation, followed by gel isolation, and then going through deep sequence, library construction and deep sequencing. That way, you are able to know where those specific RNA binding protein bind in order to understand their function in the genome. This technique is very nice, but difficult to implement. There are many technical glitches. So I managed to convince an incoming student, Yuan Xue, his picture is on the top right, to develop this protocol for us to utilize in the lab. And then we have to, at that time, have to use a robust antibody. So this is a more or less arbitrary selection for this antibody called against the RNA binding protein PTB. This gene was discovered, in fact, by Phil Sharp at MIT through cross-linking identification of RNA binding protein that bind to specific sequence important for pre-mRNA splicing. In this case, they used adenine 10 RNA synthesized in vitro, incubated with a nucleic extract, and plus minus ATP, and you can see there was a band showed up. And then this band was later categorized to bind to sequence that enriched with pyrimidines. That's why the protein is called pyrimidine track binding proteins. And then later on, it was cloned, categorized that these genes contain this protein contains 4 RNA binding domains contributed to their specific in RNA interactions. This protein was initially thought to be a splicing factor, and a year later it turns out proved to be wrong. They are involved in regulated splicing rather than have a role in the core machinery of the spliceosome. So this antibody is very robust. We therefore choose to develop our technology. And quickly we accomplished so, showing that this protein involved in regulated splicing by function as an enhancer or repressor, depending where do they bind. If they bind to the franking constitutive exons, splicing signal, they enhance the inclusion of this alternative exon in the middle. Otherwise, if you bind it to surrounding this alternative exon, it suppress this exon for inclusion, and there's no such a binding bias in constitutive exons. So that was published. So once we have this information, there are many other progress made in the field. Particularly, we learned that the PDB is not just one protein, it's a member of a family of protein consists of 3. One, PDB expressed everywhere except the neurons, and its cousin called MPDB only expressed in neurons. And there is a third member which is only expressed in T and B cells, not a topic today. As you can see here from the immunostaining, PDB red stain every cell except these neurons positively stained with NUN. This is a hippocampus image, I believe. And then conversely, PDB co-stained with GFAP, which is the marker for astrocyte. And this is the section of spinal cord. You can see PDB expressed everywhere except the neurons, but the neuron is positive stained with PDB, MPDB. So this PDB, MPDB show a temporal regulation in gene expression. And PDB in neuroprogenitor cells remain high, just like non-neuronal cells. And then upon neural induction, it goes down and remain low. And then doing this downregulation is transiently induced MPDB stay there for neuronal induction and followed by decline to stay at a relatively lower level in mature neuron. So this is a part of the neurogenesis from the perspective of RNA regulation point of view. Now we understand, after we know the genome-wide binding profile, this is the alternative exon found in PDB, MPDB, and then PDB binds to the pyrimidine track regions in the front of this alternative exon. So this exon is called exon 10. So in the presence of PDB, then you can suppress this exon, cause this exon to skip, then you run into a premature stop codon, then there's no protein produced. Once you deplete the PDB with shRNA, sRNA, whatever it means, and then this exon will be induced, then you start to produce full-length PDB2 or MPDB, also called. Okay, and now we understand the mechanism quite well. And then in order to study the genome-wide function of this MPDB, we have to get rid of PDB. The problem is that the MPDB is only transiently expressed and then disappeared. So every time we have to do this kind of a treatment and followed by a biochemical experiment, which is very tedious. So my students get tired of those process and they try to engineer the cell line to stably lock down PDB in order to have a stable expression of MPDB to provide himself for biochemical experiments. He did that. And then ran into a problem right away. He tried to do that in HeLa cells, and the HeLa cells refused to grow. Immediately upon you hit the cell with shRNA against PDB. And he has no choice but to leave the cells in incubator with hope some cell will grow up, then he has a cell line to work with. And it never happened. And this, but 2 or 3 weeks later, he found the cell completely changing the morphology. initially we thought is the contamination by mycoplasma or other things or fungus. And then later on, people pointed out to us, this look like a neuronal morphology. And that's how odd, because nobody can even see HeLa cell has a capacity to become neuronal-like cells. But we thought this is surprising or setback might be a source of interesting phenomena, so we decide to pursue in terms of the mechanism. We test multiple cell type, and turns out every single one we had a hand on by depleting PDB, every single one changed their morphology to neuronal-like cells. Okay, this is listed here. And eventually we understand the mechanism why a PDB depletion has such a potent effect to convert cell into neuronal-like cells, which is due to its another function never known before, by binding to 3' UTR of genes that either enhance or blocking a microRNA function. In this particular case, in the most case, PDB binding site turns out to be the same size targeted by specific microRNA. Therefore, the RNA binding protein compete targeting by this microRNA risk complex, the microRNA complex. So as a result, if you remove PDB, the microRNA will bind better and cause the target RNA to be degraded. And conversely, PDB can also bind a region that prevents the formation of secondary structure. So when you get rid of PDB, this secondary structure formed will prevent the formation targeting by microRNA. So therefore, in the presence of PDB, microRNA can target better. That way is promoting microRNA. That's representing the Myler class of actions. In the biological context, we understand, so this is a shortcutting into the final model from that paper published in 2013 in Cell, that this is a little bit complex, and let me walk through this. After that, it will be easier. So 2 decades earlier, we know there is a major suppressor complex called REST that prevented the expression of numerous neuronal specific genes. Many of them are TF, lineage specific transcriptional factors. And it turns out one of them is also suppress several major neuronal specific microRNA, in this particular case, miR-124. miR-124 turns out directly cause the degradation of multiple components of the REST. SCP1 is the PO2 phosphatase, co-REST is the cofactors, and the lysins. And then normally, if you turn lysins on, miR-124 will get rid of the REST. Reduced REST will further induce the miR-124, then the whole thing will spinning around, eventually cause the de-repression of many neuronal specific genes. But in non-neuronal cell, this loop does not get activated, mainly because the level of microRNA is relatively low, and even though it's accidentally induced, it's potently blocked by PDB, so that there is a break there. Okay, what's interesting here is that the PDB itself is targeted by the same microRNA. Therefore, when you remove PDB, whatever microRNA can target the REST further induce the microRNA. This microRNA further get rid of PDB and then become more efficient in getting rid of REST. So these two loops get connected to each other. In this loop, in this engine, if you may say, that PDB is almost like a jump cable. Once you get rid of it, it becomes self-enforced to get rid of more. So that's why you can just induce the depletion, and the rest of them will take care itself with using entirely the endogenous cellular programs. So this is the major advantage we explored later for converting neuronal cells, non-neuronal cells to neurons. Okay, so once we accomplished that, we realized that most of our study were performed on mouse cells, mouse embryo fibroblasts. Now we want to test on human cells. And then we take one of those human cells, very old human cells of human fibroblast, and treat it with PDB under the optimal experimental conditions. and we got almost essential every cell become a neurons in vitro. Surprising to us is that none of those neurons show any activity. They just look like a neuron morphologically. And later on, we found this is because in mouse embryo fibroblast, once you get rid of PTB, MPTB will be induced, followed by decline. So the neuron become matured. But this is a lot happening in fibroblast from humans, okay, and other cell type, other human cells. And sure enough, we examined the mechanism. Turns out all key transcriptional factors are induced when you deplete PTB except one, brain2, which is a mature neuron associated polymorph factors actually discovered by Jeff Rosenfeld, our neighbor and collaborator and friend. Okay, and if you get rid of PTB and MPTB, this gene get induced. And sure enough, if you do a sequential PTB depletion followed by MPTB depletion, and you get mature neurons. And you cannot co-deplete them together, a cell will die. So that's why we're talking about this intrinsic neuronal programming that takes its own course and reprogramming in sequential fashions. And similarly, you can overexpress brain2 and in its 3' UTR to express shRNA against PTB. Now you can turn this into a mature neuron that express every possible mature neuron markers we examined that also show many typical neuronal activity like action potentials and sodium and potassium currents. Okay, so once we understand this mechanism and be able to turn non-neuronal cell from both mouse or human origin efficiently into neurons, our next question, this is just summarized to you. There's a neuronal induction loop which is controlled by PTB. Through a break, there's a rest microRNA circle. And then this MPTB turns out to suppress brain2 expression. Brain2 activate the mirror line. Mirror line then further get rid of MPTB. So that's once this loop is on, and then you have a mature neuron. In mouse, these two loops are connected because high abundant microRNA expressed in mouse cells. But humans, you have to sequentially turn them on in other cell type. Okay, after we elucidate all the mechanisms, we next wonder, can we do that in the brain? And in the brain, one of the obvious choice for cells to become a neuron, to turn them into a neuron, is astrocyte. This is because there are several major cell types in the brain. One of them, of course, are neurons. Others are oligodendrocytes, which form this myelin sheet to protect the neuron axons. And NG2 cells, but it's very low abundant microglia, macrophage type of cells in brain, not part of the nervous neuronal cells. And then that's left to this astrocyte. But the astrocyte is intriguing in several aspects. A particular one is a highly abundant cell type, almost equal in number to neurons. So that's widely available. And secondly is that these cells can become quiescent, from quiescent to become proliferative, especially whenever there is a neuronal damage. So we thought that this would be a great advantage we can take for re-engineer astrocyte into neurons. And before we do that, we always go through the mechanisms to see what's look like in those regulatory circuitries in astrocyte. So illustrated here, this is a diagram showed earlier in non-neuronal cells, for example, miR-124, just like in the fibroblast, express low level in mouse astrocyte as well as in human astrocyte, while the neurons are expressed at a higher level. And this is in contrast to neurons, that this microRNA get induced in get induced, and then so this is a brain2 levels. Was interesting in the high level brain2 and high level miR9 as well. So in astrocyte, what we found out is that there's neuron 9 is highly expressed, neuron 120, brain 2 are highly expressed, but the neuron 24 is just like a fibroblast. And then this is the same thing is true in human cells. That means these two loops are distinct. The first loop in astrocyte resemble the non-neuronal cells, but the second ones resemble neurons. And then this is because astrocyte actually share the ancestor with neurons, being radioglial cells. So that's why there is a related gene signature, but astrocyte are a lot neurons. This indicates that we can just turn on the first the checkpoint, and then the astrocyte should have a potential to become neurons. And sure enough, by testing these ideas, we get rid of PDB in human adult fibroblasts. You can turn on MPDB, but never going down. That neuron is not mature. And then with astrocyte from either mouse or human, you induced MPDB, and the MPDB naturally going down by itself and ripping at the lower level, indicating they may become mature. And sure enough, we categorize this is indeed the case. And then this has become so potent and so efficient in our hand. Okay, so this is a PDB lockdown, and you can see the high efficiency. You can see recording the activities. So we have done a series of experiments to categorize those in vitro converted neurons. And then next, if we can do everything so well in vitro, we would like to test it in the brain. And this is our targeting vector. We use AAV as a vehicle to deliver our genetic payload. In this case, we have a RAP for lineage tracing, and followed by shRNA against PDB in the 3' UTR. And in the front, we place expression stop that is flanked by LOXP. As a result, these transcriptional units are completely silent in other cell type, except if you inject this AAV into a transgenic mice that express Cre recombinase from astrocyte-specific promoter, GFAP promoter. In that case, this cassette will be deleted, not permit the expression of RAP for lineage tracing and SHPDB for converting the cell fate, changing the cell fate. And we have done many experiments to categorize these vectors and show this perform supposed way. And we found initially most of the cells infected are a lot NUM positive rather than a GFAP positive. Most of the cells infected by the virus that express the red fluorescence protein. But this cell body undergoes progressive conversion into the neuronal feature that stain positive for NUM. In our experiment, we focused on this region called substantia nigra, which is the origin for the birth of a dopaminergic neuron, relevant for our later studies. As you can see here, a large fraction, about 30-35% of neurons converted become dopaminergic, stain positive for DDC, which is the gene important for dopamine transport, as well as tyrosine hydroxylase, which is the important enzyme for dopamine biogenesis. And then this neuron express very specific sets of channels called HCN channels. That the early one, we don't see any of those activities, but later on we see those channels. These channels can be blocked with cesium chloride, characteristic of a dopaminergic neuron. And then this maturation also become evident very clearly with the action potential, initially very infrequent and later on become very frequent. And then this firing is typical of endogenous dopaminergic neuron. And we have also many other criteria to let us to believe that the newly converted cells look like endogenous dopaminergic neurons. And what's interesting here is that we inject our AAV in the midbrain that can be converted into a Th positive dopaminergic neuron. We did a similar injection in striatum, is where a dopaminergic neuron send their axon to to be connected with a GABAergic neuron. And turns out the efficiency is very similar, but most cell converted a lot dopaminergic neuron instead. Most of them correspond to GABAergic neuron. And yet again, in cortex, and most cell become glutanergic neuron or other type. So this is clearly indicating that a different brain region, astrocyte in different region, have a tendency to become a neuron that related to endogenous neurons in that region, so-called regional specificity. There was a lot of a mechanism we elucidated. The one mechanism is the intrinsic mechanism within the astrocyte. This here we are comparing the cortical astrocyte with the midbrain astrocyte. This set of factors, TF, transcriptional factors, are typically expressed in dopaminergic neuron, and they show higher basal level in midbrain astrocyte. And upon PDB treatment, they show much higher level of induction as well. But however, this does not fully explain the high percentage of dopaminergic neuron generated in vivo, indicating that the local environment may also made a substantial contribution to this regional specificity. And not only we can see the progressive maturation of dopaminergic neuron in nigra region in the cell body regions, but also we can see their projection of their axon to striatum. In this case, you hardly see any red fibers in 3 weeks, but gradually you obtain more and more fibers, and eventually to a higher level, which will be which is quantified here compared to endogenous ones. And then by lineage tracing, we further demonstrate that these new neurons somehow get integrated into the endogenous circuitries. Here we inject a green ritual bead into striatum regions, and then 2 days later examine this ritual bead in this substantia nigra regions in the cell body. As you can see here, there's a green bead labeled endogenous dopaminergic neuron. This is all done in wild-type mice, as well as newly reprogrammed neurons. This is done in the again in wild-type mice. And then in this nerve terminals, you can also see this red fibers form synapse, as indicated by this pre- and post-synaptic markers for neurons. Then if everything is done so well, then we can then start to ask questions whether or not we can test the functional benefit of these new neurons converted from astrocyte in the disease model. Here we used this chemical-induced Parkinson disease model that by using in which we can use 6-hydroxydopamine, which is the analog of dopamine, that specifically get trapped into a dopaminergic neuron that produce a high level of RAS in the end, damage or cause the death of a dopaminergic neuron. So this chemical model, we need to make it clear this is not a disease per se, because it does not show there's a progressive dysfunction of dopaminergic neuron. Rather, it potently, acutely kill the dopaminergic neuron, therefore reflecting the end stage of Parkinson's disease, which is the loss of dopaminergic neuron. And then this is a little bit complex here. And then so this is midbrain regions. Left side is intact side, and the right side is where we inject these chemicals, which dramatically reduce the level of dopaminergic neuron, Th positive dopaminergic neuron. And in the same time, you see more astrocyte get induced, much more. And then after reprogram, you see many of them become a dopaminergic neuron again. Okay, and then this is on the top of this substantia nigra in the striatum region where the dopaminergic neurons send their axon to, and you can see in the unleashed side there are a lot of those Th positive green fibers, and then upon lesion, you lose those fibers, and then after reprogramming, you restore. As quantified here, restored to the wild type level of 35% levels. Then we can also we also determine that not only you can integrate it and then get connected there and then restore the lost dopaminergic neuron, you can also regain this activity-induced dopamine release. In this particular experiment, we take live animals and stick electrode probe into near the substantia nigra regions, and then record using a carbon fiber electrode to record the dopamine release. As you can see here in the intact site, we have a normal activity-induced dopamine release, but in the lesion site, the activity is drastically reduced, and then you can largely restore this one. And you can take these live animals and cut their brain and measure on their brain slice, you get essentially the same results. And then this is reassuring that everything looks like a wild type levels. At the behavior levels, then we use the two type of assays. One is a rotation assay, one is a nimble touch assay or cylinder assay. As you can see here, this is a disease. Lesioned mice that spin in the clockwise fashion, and the same mice after reprogramming, it does not show such a movement bias anymore. Okay, as quantitatively as we can see here, this is a bit more complicated. So this is a rotation assay, and this is the nimble touch assay. You can see here, wild type, there's no bias. And equal 50-50 use of left and right nimble, and then the lesion mice showed this disease-related motor phenotype, and then after reprogramming, this phenotype gradually disappeared, reached to the wild type level, and same thing is the case in these behavior assays. The question always raised here is whether or not you induce new neurons and the new neuron repair the existing circuitries, so on and so forth. There are a cascade of events which takes 3 months to accomplish. And to demonstrate that the whole behavior is actually controlled by our engineered neurons, and we did the following chemical genetic studies. In this case, we replaced RLP with engineered GPCR that can be turned on by a chemical called CNO. So then you reprogram this astrocyte, that astrocyte will contain this GPCR. Then you can see their phenotypical recovery, but however, which takes 2 to 3 months to accomplish. But once you inject this chemical CNO, it will bind and activate this GPCR to cause a hyperpolarization of the cell. As a result, you can diminish the action potential in 1 hour. So basically, you lump up, but the cell is there. And then if you later on, after this drug is metabolized in 2 days, 2 or 3 days, and then you are supposed to recover your signal, the effect. So that means everything is reprogram based. So indeed, that's the case. That if you lesion, you have this biased touch assays, and then after reprogram 2 months, then you restore the phenotype. And then if 1 hour, if you inject the chemicals, the phenotype appeared again. And after is metabolized, it retained the phenotype. And if you do not engineer the reprogramming, you basically do not have such a behavior benefit or response. So this is demonstrating that it is the engineered neuron that is doing the job to correct the phenotype. Of course, other neurons may also become more efficient in that process in the coordinated fashion. So far we've been using AAV, which is a genetic way of deliver a genetic payload. We just wonder because in our mechanistic understanding, the PDB looks like a jump cable. Once you started your engine, you don't need it. So we said that this might be a great advantage for using this ASO, antisense oligonucleotide pioneered by our colleague Don Cleveland Lab. And in this particular case, ASO will bind to your RNA. This is a DNA-based large molecular drug, not bind your target RNA and then use endogenous RNase H to cleave your RNA to cause downregulation. And sure enough, with PDB-specific ASO, we can turn astrocyte in vitro into neuron as well as in vivo. And at the benefit level, you can see the correction was PDB ASO, but not with the GLP ASO. So this is provide a proof of concept experiment that we may use the non-genetic material and through this dosage controllable ASO strategy to turn non-neuronal cell into neuron to replace the lost one in specific disease settings. In summary, this paper was published 2 months ago by now, how time flies, in which we demonstrate the modulation of a regulatory RNA circuitries enables efficient reprogramming of astrocyte to functional neuron. As you can appreciate now, it's purely from a basic science, try to understanding some regulatory gene expression paradigm that ended up leading us to making new discoveries. And then we show a single dose of either AAV or ASO anti-PDB agent is able to regenerate dopaminergic neurons to reconstitute the nigra striatal pathway. So this is very key, reconstitute the lost neuronal circuitries rather than just providing new neurons that secrete dopamine. And these reprogrammed neurons are able to efficiently reverse a chemical-induced Parkinson disease phenotype in mice. And in fact, we have been keeping our both a group of lesion mice as well as a reprogrammed mice for almost lifelong, for 2 years, and we still see the phenotype recovery, indicating that the new neuron gained lifetime benefit that as healthy as the normal mice. And because we found our approach appears to work in multiple different brain regions that generate neurons related to endogenous neuron, we feel, but have not yet tested, that this approach may be broadly applicable to the treatment of different type of neurological disease, neurodegenerative disease. So I would, this is prepared for your questions, but at least out here that I can elaborate more. Obviously, we are not stopping here. We need to test our approach on genetic PD models as well as extension to other disease models such as Huntington's and Alzheimer's. And we need to develop a strategy to inactivate existing disease mechanisms. For example, people are also curious. You make a new neuron from the endogenous in the brain that has already some other defect already. Those defects may make the new neuron less healthy or die quickly. The question is, can we do both? Not only make a new neurons and make them resistant to the existing disease mechanisms. We do have a lot of idea to do that. And we everybody knows that the cell reprogramming due to the increasing stiffness of epigenetics in the genome, that cells become more and more difficult to reprogram. And now we believe we have a strategy to rejuvenate astrocytes before we reprogramming them. And again, this takes a full advantage of astrocyte that can be induced from quiescent to proliferative state. That will allow us to do that trick. And of course, before eventual applications, we need to determine the potential side effect, both as a result of astrocyte depletion, although we argue that might not be so bad because astrocytes get proliferative, doubled multiple times, and then we just convert a small fraction of them back to neurons. Or due to the too many neurons generated, or wrong targeting of those new neurons, that may cause problems. But we can know that only by carefully check the potential side effect. which, by the way, is the same problem also. There's too many neurons or too much non-specific targeting may be associated with also stem cell-derived neurons. And finally, and most importantly, we need to demonstrate the efficacy of approach in non-human primate models before we can embark on clinical trials. And since the publication of a paper, I received numerous phone calls from patients who are so passionate that they are willing to try anything. And somehow this approach appears to quite appeal to them. Maybe lie in its simplicity just by getting rid of a single genes. Finally, I acknowledge there's two key guys in the lab. First, Yanchao Xue, who is a postdoc, discovered these mechanisms. And then by the time he was ready to leave the lab, we are lucky to recruit a new one, Hao Chen, who is the first author of the paper. And he was trained as a hardcore electrophysiologist in neuroscience. So as a result, he single-handed developed the whole paper. And that, of course, took him a couple of years to do that. And the paper contains almost like 200 panel of figures. So it's remarkably productive. We have one set of experiment which is listed activity-induced dopamine release, just completely beyond our expertise. So we ship our mice to Peking University in China for two collaborators to help out with that experiment. Because my lab is not a neuroscience lab, even though we try to learn along the way, and we have learned that for 10 years, but I still feel I'm an outsider. Fortunately, I have a great collaborator to ensure the data quality as well as the interpretation of those data. In this particular case, is Don Cleveland, who gives us a lot of guidance into disease models, and Bill Mobley, who is the neurologist who interpreted all our immunostaining data and other type of data. And Steve Dowdy is the molecular biologist, a chemist that provide expertise for us to develop an ASO-based approach. So together, we are very grateful to have an opportunity to work together, and UCSD is so rich in such expertise as well as a collaborative environment for us to do many things that are beyond our expertise comfortable zone. So this is the feature of UCSD. We hope to continue that tradition. So I'll stop here and answer your questions you may have. Thank you very much for your attention.
One of the earliest questions that came in is, do you think any of this applies to the peripheral nervous system?
The peripheral nervous systems, if you talk about the motor neuron, we have a very, we don't know. The confidence level is not very high to hope to generate motor neuron that can grow a meter long. However, that's not to say we cannot generate interneuron that might be helpful. We have not made a major effort in that area yet. In fact, there are local experts in that area who would like to start some collaboration. We'll be very happy to engage such a collaborative activities in the near futures.
One of the questions was, when you do this, converting astrocytes to neurons, are you compromising astrocyte function in the brain?
Right. So we mentioned that, I mentioned that at the end of my talk. Astrocyte, of course, is very abundant. We know astrocyte have many roles in neuronal functions. And when it comes to disease, it also have a role in protecting the neurons. Okay. On the other hand, the astrocyte is also known to have a role in further damaging the neuron, especially when it's become over-proliferative and then form a scar that causes further neuronal damage. At this point, we just don't know what kind of adverse effect we may create it. The hope was that we only convert a small fraction of them out of those proliferative astrocytes. So therefore, the damage is not as severe as supposed to be. And we are also thinking about the idea, because astrocyte can be divided into A1 or A2 type, A1 being more harmful to the neuron, A2 are protective. So if there is a way to selectively target one type but spare the other, now be a plus. But astrocyte biology is very complex. We are still in the learning curve, and we are consulting experts in the field to guide us. And ultimately, it is to test whether or not there is any side effect or major side effect by turning the astrocyte into the neurons.
Great. Another question is, how unique do you think the PTB1 knockdown effect is? For example, have you ever tried any other RNA binding proteins like HER, ILAV1, or anything else?
Yeah, we have not tried systematically. There are RNA binding protein, but many RNA binding protein, you have to identify RNA binding protein because if the goal is to change the cell fate, you have to identify RNA binding protein that undergo such a change in expression or localization, whatever property associated with that process. And so far, we have only focused on PTB. Does not mean there's no other things around or in other systems that we can utilize a different type of RNA binding proteins. But MPTB, because it's silenced in the beginning, you cannot manipulate them too much. But however, if you, even though it's dramatically reduced in mature neuron, you cannot get rid of them. Otherwise, the newly converted neuron or neuron will die in a month or two. So even in the, if you get rid, that's why if you get rid of PTB or MPTB genetically, you basically obtain no neurons. I'm sure some people will ask that kind of questions. So now we are just basically tilting the balance a little bit in the cell to encourage them to differentiate into the neuronal lineage. But everything else, just let the cell to handle in the progressive or stepwise fashion.
Okay. Another question. Why do you think the astrocytes convert to dopaminergic neurons in vivo and not other types of neurons?
We do get a mixture of neurons. It's not like only dopaminergic neurons. We get about 35-40% dopaminergic neuron. But that percentage is dramatically higher than other regions. For example, in cortex or striatum, we only get less than 1% dopaminergic neuron. As I explained in brief, that this so-called regional specificity is likely contributed by both cell autonomous program as well as a local environment, just like a tumor environment. The cell autonomous program, in principle, has been demonstrated recently last year by Chris Glass lab using macrophage. Even though all those macrophages look the same, but macrophages in different regions have their enhancer pre-wired. So in the other words, the 3D genome are different. That allow utilization of different enhancer to activate different sets of genes that are more particularly prone to reprogram those gene expression to encourage those cells to become neurons related to the endogenous neurons. Okay, so this is become all of a sudden become a 3D genome problem. So if you really want to understand the mechanisms.
There's two questions that kind of get at the same, try to get at the same point. One of the questions was asking you whether you could elaborate on the effect of local environment on the conversion, and related to that was a question that there are likely some regional patterning factors that are secreted by neighboring cells that help direct the astrocytes into dopaminergic neurons. And did you see any reduced efficiency in conversion in the disease model compared to a wild-type model? So questions getting at what do you think is going on in the microenvironment?
Right. I do not have too much to say about the microenvironment at this point. What's interesting here to note is that people have developed all kind of a protocol to convert or strategy to convert non-neuronal cell to neuron. But under those in vitro conditions, you need to not only engineer your key reprogram genes, but also change the mediums like N3 medium that has drastically reduced the serums. You add NGF or many other trophic factors to encourage them to become a neuron and become a healthy neuron. And of course, in vivo, you cannot do that. You cannot inject the whole thing along with a cocktail. So that's entirely depends on what's there in vivo. Okay, so in vivo, our guess is that many trophic factors like BGNF and those kind of things, which has been implicated, play important role for conditioning dopaminergic neuron. But other than that, we don't know. Okay, so that's the number one question. The real answer is we don't know what's going on. And then, so the next question is that whether or not you show any difference in normal mice versus diseased mice. So far, we did not see any difference, but that's not to say there's no difference. Especially our model is a acute model, which is why we need to develop a genetic model to allow disease progression in the more lateral related pace. At that time to see if there is a local environment that get damaged, but so that you may mess up the different type of neuron being generated. That said, we can also find a different way by using key lineage factors to enhance the process. We try not to use that at this point because the more you express, the harder to implement later if you do that for clinical trial. For example, people use lineage factor to convert dopamine neuron. They have to use 4 different factors to do so, or sometimes 6. So that's a little bit, a lot.
Okay. The next question is a double-barreled question. The first part of the question is that young mice might have a higher regenerative capacity. Have you tried this approach in aged mice? That's the first part of the question. The second part is, have you ever tried converting microglia to neurons?
Okay. Second question is easy. We have not. However, other people try to use PTB to convert oligodendrocyte to neuron. They are successful and quite efficient. I don't understand the rationale for such experiment. By reading the paper, which was published in Molecular Therapy, they just want to use this as a way to test their trophic AAV developer in that lab. Okay, so that's the second question. The first question is aged mice. And in fact, during the revision process, we were suggested by the reviewer to do such experiment. And we thought it's never going to work because actually very few people have tried those kind of things. Most people just standardly use this standard model is a 1 or 2 year old mice in this case. Okay, so but the reviewer say if this thing is going to be applicable to human, eventually you have to deal with these aging issues. So we therefore did such experiment on 1 year old mice. So started with 1 year, you treat with 6-hydroxydopamine, and 2 weeks later, you wait until the stable phenotype, and then you go through reprogramming for 2 months or 3 months, and then recording. So by the end, it's almost like the end of the lifespan of the mouse. So it's 1.5 or longer years old. Okay, so we did that. So initially, we were anticipating total failure, and remarkably, one of the assay works just great, which is the cylinder nimble attached assay, showed the fully reprogramming. And we're amazed by seeing those results because in our hand, the reprogramming efficiency reduced to about 10-20% on aged mice. But there is a strange phenomenon here that even though we have a very limited reprogramming generated a reduced number in substantia nigra, but somehow the density of Th positive density fiber get a little bit higher than just like a correspondence increase of cell body. And then that further translated robust dopamine release, about 80%. So it looks like something going on endogenously turn into the whole system better with the newly generated neurons. We still don't know do not understand the mechanism yet. Nonetheless, people are saying that the new neuron can help repair the damaged ones in other type of transdifferentiation studies. And then the other assay, which is a rotation assay, did not work. We could not get a statistical significant experiment. Some mice work, other mice messed up. And we realized that this is related to this unstable phenotype in aged mice. Actually, the aged mice spin around much faster for some reason. That makes it difficult to record. So we initially report back to the reviewer saying this experiment are inconclusive. So we would like to propose to leave it out and let us do more studies to understand. But the reviewer have a opposite view. They said this is exactly what we predicted. This is such an encouraging result. Not only you have to show it, you show that in the main figure. So that become a part of the main figure, even though all those data get stuck in tiny little font, the smallest possible, in order to fit the page limit. Okay, so that's in the paper, not data. But we need to do more on that.
Okay, the next question is also a two-part question. One was, did you ever try the reverse experiment where you did the transdifferentiation first and then do the lesion? And would you predict that there'd be any difference there? And then the second part to that question is, are the transdifferentiated astrocytes replenished? In other words, when you convert the astrocytes to neurons, what happens to the pool of astrocytes? Do they get replenished, and when does the transdifferentiation stop?
Okay. The first question is whether or not we have done the reverse experiment. I don't think we have done that. In the other word, reprogramming followed by lesion. We have not done that. What's interesting here, this is related to a related question is interesting, is that whether or not you can generate a neuron and the neuron has some track to follow to target into the right region, at least a part of them. Okay, so we initially did in the wild type mice, they do. And then later on, we did lesion mice with thought some of those tracks may get lost. Eventually, if you have too many things damaged, it may lose such a track. But it turns out that's not the case. Not even though you wipe out 95% or 95% of dopaminergic neuron, the new neuron can still find a way back in a similar way. We could not see any difference. So the short answer to the question is that no, we have not done the converse way, that wipe out the neuron after reprogramming. And then the second question is related to, could you remind me again?
Yeah, what happens to the pool, what happens to the astrocyte pool, and when does transdifferentiation stop?
Right. So there's a transdifferentiation, we are using the AAV to do the transdifferentiation. And then the AAV do in the initial infection, whatever cell you get into, a fraction of them become a neuron. And after that, either become a neuron or the cell remain as an astrocyte. So in the other words, in our hand, when you have a lesion, the astrocyte become amplified. But without lesion, the astrocyte do not. But there are abundant endogenous astrocyte. So, however, after reprogramming, we do not see any further depletion or amplification. Looks like they remain the same. They don't change anymore. So in the other words, it's not like you make a new neurons that the astrocyte become coordinated, increased in order to protect the new neuron. Probably there are a sufficient amount of astrocyte there in the first place that you don't need to generate more of those. And most of the time, astrocyte become proliferative in response to neuronal damage or in response to some stimuli like LPS. In fact, that's the property we like to explore to encourage their reprogramming. But after that, we don't see much of the proliferation of astrocyte or depletion of astrocyte.
Another question is, can you comment on any of the other genes that that might change during astrocyte conversion, such as SOX2, neurogenin, NeuroD? Do you have any knowledge about those?
Yeah, we did. In fact, in the paper, we did extensive RNA-seq studies. Initially, they look like astrocyte, but a different region of astrocyte show different. But even the same batch, different batch of astrocytes show difference. So there was an enormous of heterogeneity among astrocytes, we noticed. What's interesting here is that once you convert them into neuron, those gene expression is almost like a replicate of the same sample. There's no difference anymore. They all look like the same type of neurons, mixture of neurons, okay? So they're almost identical. In our early experiment, of course, in those induced genes or repressed genes, many astrocyte-specific genes get reduced or diminished, or many neuronal genes get induced. However, we never see any induction of proteopotency genes like SOX2 or SOX9 or other things. This is because our cells, unlike any other transdifferentiation systems, that there are a few cell dividing before differentiation. In our case, we can convert them into neuron without any cell division, or only if they are not arrested at G1 cycle, they may go one round. That's it. So in the other word, there's no cell division. So the cell never do not go back to a pluripotent state before differentiation, differentiation into neurons. So that means there's nothing, no pluripotent genes being induced. That said, once you get rid of PDB, essentially every single factors being examined and tested for cell fate change to convert a fibroblast to neuron has been induced. And the only distinctions we have is this brain 2. That's the only exception. That's the only exception happening in human cells, not in mouse. Mouse, the whole slew of factors are induced laterally. And it turns out the vast majority of them are targeted for rest. So in the other words, the whole circuitry has to be related. Otherwise, you cannot just change the neuron with a completely different gene, distinct gene signatures.
Okay, well, I think we're down to our last two questions. One question is, what do you know about what normally happens to PTB in disease in general, or neurologic disease in general? Do you have any knowledge about
neurological disease? The only thing I know is that the PDB get enormously increased in GBM. And actually, if you deplete PDB using AAV or other things, you can potently stop GBM from growth. And so that's in neurological disease, because this is a lot expressed in neurons, obviously does not. And then in AD or many other diseases, you normally see when you do a bulk studies, PDB get induced. This is most likely related to the proliferation of astrocyte in those diseased brain. Okay, many most likely due to the astrocyte, but we don't know yet. In the limited published study is based on astrocyte activation.
Great. And I guess the last one is more of a comment, and the comment was that it appeared that the axons, your axons, new axons, grew amazingly fast. Do you have any comment on the speed with which the new axons grew?
No, not amazingly fast. This takes us 3 months. And we calculated the growth rate is about 4 millimeter per day. That's what it takes. And it turns out that if you measure the growth rate of axons in test tube, in petri dish, is the same rate, regardless if the human or mouse cells. So indicating that the growth rate is intrinsically determined, is not influenced by outside conditions. And then that's translated into a challenge in a way that if we need to see this procedure to work, we have to, in the monkey or human brain, the axon has to grow longer. And then that's the challenge. So the question is, can we do that or not? By estimate, in humans, you need probably half a year to do that, or even longer.
All right, I think we've addressed all of the questions. I'd like to thank you for giving us a great talk and for everybody for attending Southern California Stem Cell Consortium under unusual circumstances, to say the least. We also thank the UCSD Alzheimer's Disease Research Center Neurodegeneration Seminar Series for joining us in this. And I guess we'll draw to a close. Have a very safe and productive day. Bye-bye.
Bye. Thank you.